right, everybody. Thank you for joining us once again, where my very learned and also very, very welcome guest is Dr. Jacquelyn Fede. Her role today is in translating my scientific gobbledygook to the best of her ability into English, so that y'all can get an idea of what on earth it is I'm talking about with this thing that I'm always banging on about with noise to signal ratio, or signal to noise ratio, if you like. So I thought we'd start the discussion with Jacquelyn telling us really about these two metrics that are used in statistical inference. One's called the standard deviation and the other one's called a standard error. So what I'm going to do is basically shut up, because I talk too much anyway, and put her up on her hind legs. I'm assuming your gender, by the way. That's very naughty of me, isn't it? You might be an attack helicopter.
You know, ever since you've said attack helicopter, I want to be one.
Yeah.
But
well, you can.
I don't latch on to trendy labels. I kind of just go with I've got a vagina and a menstrual cycle, so
right. Oh, well, let's assume. Let's assume, shall we? You know, I am an attack helicopter, and my pronouns are.
Oh.
And that's how to pronounce them correctly. Otherwise, it's hate speech. Frankly.
I might have to take my sweatshirt off to make your proper noise. I apologize. Excellent. I didn't come prepared.
Well, next time. Okay, anyway, so tell us all about standard deviation, standard error. What on earth are these numbers? Where do they come from? And how are they used appropriately and otherwise? All yours.
Oh, thank you so much. Well, standard deviation, let's start there. So anytime that we want to look at a population or make any kind of claim about a population, it's virtually impossible to gather the entire population and ask every single member. So in science, when we are trying to think about our population, we are sampling from it. We are taking a group of people from that population in hopes that the sample that we take is representative of the population at large. So let's keep that in the back of our minds. So we've got this sample, and we're going to have measures that we care about when it comes to this sample. And for any of those measures that we care about, we're going to probably look at a way to summarize it for our sample. And one of the ways that we commonly do this is the mean, the average. But the mean on its own can be pretty meaningless. You might have a sample where the mean is 50, and everyone in that sample has scored on some measure between 48 and 52, right? And that's a very different mean a very different sample from one that has a mean of 50, but everyone has scored either between 0 and 20 or 80 and 100. And no one in your sample actually falls at the mean at all. And actually, you shouldn't be using the mean as your main metric there anyway. But there's going to be a spread. There's going to be a spread around that mean, because it is true that not a single person in a sample could actually fall exactly at the mean that we've calculated. And how we get a grasp on how wide that distribution is around the mean is this measure called the standard deviation. It's essentially the average distance from the mean. So we might say that our standard deviation is 2. So 50 with a standard deviation of 2 is meaning that 95% of that sample is going to fall within that 48 to 52 range. We want a small standard deviation. Deviation is the noise when we're talking about the sample. The mean is the signal. If we have a huge standard deviation, it is unlikely that any change we see in our mean is going to be meaningful because it's likely to fall within that noise range. And so we can't say with confidence then that a mean that moves up 2 or 3 points within a standard deviation that is 5 or 6 means anything but random noise, random error. It's something we would expect to see within the average distribution of our sample. So it really undercuts our ability to make good quality claims about that mean or any change in that mean, or it should signal us to not make claims about that mean so much. So that's all on the level of one single sample, right? Our sample that is supposed to be representative of our larger population. There are cases where you may truly only care about that one sample you picked out. I've done some work in program evaluation, for example. And if this group selected into the program because they wanted that program, I might truly only care about changes to this smaller group. I may find amazing findings that I might very hesitantly say, you could consider this for similar people in the population, but I'm really doing it for this group that's selected into the. In health and medical and nutritional and many other forms of research and science, we really care a lot about our population. We're sampling from the population in hopes that we can make claims about that population. And we want to be able to say this sample that I've taken, this smaller group, is representative of that population. And some of the ways we do that might be listing out. You'll often see a table of the sample demographics. Unfortunately, they don't often put that next to a table of the population demographics to show you how similar it might be in that regard. But that's one way that we often think about it. But another really great way and critical way to think about it is the standard error. This is like the meta standard deviation. So this is looking at, so I've taken my one sample and I understand this dispersion around the mean that is standard deviation. But how close is my sample mean to what the actual estimated population mean would be, or the real population, the actual population mean? And we have statistics to think about this, and it's called the standard error of the mean. And it's basically, if I drew that sample or a sample 10 times from that population, and I plotted all of those different means, what would be the average or the standard deviation of those means of the population? And that gives me an idea of how close my sample mean is to the actual population. And this is another same issue. If I find that my standard error is very large, I should be very wary, if not completely put off, about trying to make generalizations to this larger population. And I think mean and standard deviation are thrown around all the time. Standard error is less utilized, unfortunately, even in these cases when we are trying to understand, can we generalize to the larger population? It often just conclusions and inferences just go there.
Right.
I guess we'll start there, and then
brilliant.
We can.
Yeah, absolutely brilliant. I think that's a very good description. So thank you for that. So what it seems to me that what we've got here is if I have a study that I want to do and I take 10 people and put them in group A, my control group, and another 10 people and I put them in group B, my experimental group, and then I measure them for some outcome variable, whatever that may be. Mm-hmm. And I get a mean value for that particular outcome variable in those 20 participants split into two groups, 10 in A and 10 in B. And then I report to the journal that I'm writing the paper for, here was the mean or average value for output variable X, whatever that might be, or Y, actually, because X is usually your control or your time course or whatever it is. That provides the journal, if I just said, here is my mean, full stop, that provides the journal with how much information about the value of that mean? None. Absolutely none at all. Because, as Jacquelyn said, my mean might have been value 50 plus or minus 2 for a standard deviation. So to get 66% of every observation I actually made, I need one standard deviation. And for 95% of all my observations, I need two of those. 1.96, but let's say 2, just to make the math easy.
1.96.
Let's call it 2. That's near enough for me. Okay. So if my standard deviation around my mean was 2, then for 95% confidence, I have to say that my mean value in those 10 pairs of observations was 50, if the value was equivalent in the two groups. The mean for both groups was 50, but for 95% confidence, it was somewhere between 46 and 54 in both groups. And one group might have been closer to 46 in reality and the other closer to 54 in reality. But because I've just said the mean was 50 in both groups, there was no change. The journal has nothing to go on. And in fact, what they would do is they would send that paper back to me and say, provide some gauge of variance around your mean. Provide us with at least a standard deviation or a standard error or something. Mm-hmm. So then I could write them back and say, oh, fine, well, the standard deviation was 2. So that means, we're between 46 and 54 in both groups. So actually to say there was no difference now becomes quite tenuous. So we may be making a type 2 error statistically in so doing, if we were not to say, but here was the standard deviation in that observation. Was that group different from that group?
Yeah.
Okay. Now, what Jacquelyn was also then quite eloquently saying, I thought, was that, okay, fine, but how does that relate to the general population if instead of sampling 10 people, we somehow were able to test everybody that was representative of that population? What number would we get for the mean of that? So how we do that mathematically is we take the standard deviation of however many observations we made, and then what we do is we convert that using the square root of n, the number of observations that we took. And in so doing, the standard error becomes vastly smaller than the standard error of any one sample taken from that overall population, depending, of course, on the sample size. The bigger the sample size, mathematically, the smaller the standard error becomes, because the larger a sample group, the more representative it is of the entire population from which that sample was drawn. How am I doing?
You're doing very well.
Awesome. Great.
Very well.
All right.
Yes.
So here is the confidence trickery I find in statistics. I'm going to throw this out there and let Jacquelyn respond with her opinion. She is absolutely free to disagree with me if she so sees fit, of course, with the caveat that she understands that in so doing, disagreeing with me, she makes herself guilty of hate speech on my channel. I'm just joking. Of course, you can have any opinion you like, so long as it agrees with mine. But here it is. Confidence trickery in statistics. Okay, the t-test, which tells us whether two groups are statistically different from one another. Mm-hmm. Inherent in the mathematics of the t-test, the factor, the measure of variance around the central tendency, is the standard error. The same is true in the F-test, the ANOVA, the ANCOVA, the ANOVA with repeated measures, all these tests of test-retest reliability of a mean value. Every single one of them inherently in the mathematics contains the standard error, not the standard deviation. And in so doing, if we have a large enough sample size, we can find a statistically significant result because the standard error is very, very small.
Yep.
And we can say, look, these groups are statistically significantly different. Hey, presto, now I'm a valuable human being as a member of the scientific community, and I've given you some utilitarian knowledge here that you can go forth and be confident with. Aren't I a great person? Where's my Nobel Prize? Despite the fact that the standard deviation around the observations I actually made might have been so wide as to render that actual finding completely swamped by signal-to-noise ratio. And in science, and in health science, nutrition, sport and exercise science, every field of science I've ever been involved in, cardiovascular pathophysiology as well, you will find signal-to-noise ratio which absolutely swamps, so that the noise swamps the signal. Ergo, the signal, while it is statistically significant because of the sample sizes, its utility is 2/3 of 5/8 of 7/9 of bugger all. So, how do you feel about that?
Oh, I feel strongly about that. And I feel even more strong about that now, as I've seen in science a lot of machine learning techniques come about that are just there to draw thousands and thousands and thousands and thousands and thousands of random samples over and over and over. And you can essentially, everything's overpowered. It's just tons and tons of sample size, and your standard error will be minuscule. It's when I first started taking statistics classes, I remember someone saying, imagine a factory, and it's cereal, not a pleasant sight, but cereal boxes being filled, and every box gets one extra cornflake in it. In any one box of cornflakes, that is meaningless. That is nothing, right? It is practically not at all significant. There's a difference between practical and statistical significance, and this is where it shows and rears its ugly head. As all of those millions and millions of boxes go through the conveyor and get that one tiny cornflake added to them, eventually it will come up as a statistically significant difference. And it means nothing except a publication for someone funded by the cereal industry. No, except for a publication, a meaningless, useless publication.
Yeah.
And I also think it's really important to say that there are labs. I mean, a lot of research, unfortunately, is coming from a lab headed by someone with academic prowess or power who has funding and grad students and access to massive data sets. And they're just running analyses. They're just finding significant things in these massive, large data sets. And there's a whole other problem with just running tons of analyses. There's also statistical corrections for that, because the more tests you run, the more likely it is that you'll just happen to find something as well. But these samples, these data sets that they have are so enormous that you're just going to find something, and they just publish it and it's meaningless. And there's so much noise, and it's a real problem. So I agree with you there.
Excellent. Good. So you escaped the hate speech clause. Love it. Love it. So,
Yes.
And I guess that feathers quite well into the part of the discussion where I wanted to talk to you about a letter that was written by yours truly a few years ago, quite a few years ago now, actually, to a journal known as Advances in Physiology Education, which, among other things, invited commentary from various people at various levels of learnedness or otherwise, regarding standard procedure, I guess, in the science of the epistemology, I guess, around such areas, and including things like statistical inference and stuff like that. So I wrote a letter which contained 4 paragraphs at the time of writing, at the time of submission. And we can go through it for a bit of fun, because I think it makes the point. Well, I hope it does, and so did the editors of the journal at the end of the process of peer review. But of course, I only got it published with the third of the 4 paragraphs I wrote deleted in order to get it published, because I went in that paragraph so far as to challenge standard procedure, and as such, because that wasn't accepted narrative, even though they'd invited commentary on that, in actual fact, the invitation for commentary on it was lip service. They didn't want commentary on it. They wanted someone to espouse the standard argument, or at least tiptoe around a challenge to it. So my letter ended up being a tiptoe around rather than a full head blown charge through the brick wall sort of letter. Also, I was required to insert the first 3 words before the comma of this letter. I was required to insert those 3 words by my department, because I was working for a department that...
I'm looking at those 3 words.
Yes, and it says, the 3 words I'm talking about are, I had to start my letter with, as a non-statistician, comma.
Well, relative to what?
Well, relative to the fact that the university I was working at the time had a department of statistics, and I was not employed in that department.
Oh, dear.
Furthermore, at that time, this is 2008, I did not hold an advanced research degree in statistical inference, which has since been rectified. However, they said to me, well, you're not a statistician, so you need to disclaim yourself as such, even though what I've actually had to say about statistics here is absolutely unassailable, I think. But anyway, as a non-statistician, I said, I have watched the discussion on this topic with interest. The background is that there was some groups that were suggesting, heretically, that if we want to be disciplined in our discussion about scientific findings, let's go to that example of 10 observations in one group, 10 observations in another group, and at the end of our paper, we want to talk about the value of that, the statistical inference on that. If we want to talk about a difference between those 10 people and the other 10 people that we actually looked at in our actual study, then we need to describe the value of the difference between those 2 means, should one exist, using the standard deviation, because we're talking about those 20 people. And that was heretical, and it started this shitstorm backwards and forwards in this journal, in the letters, which is what prompted me to write my letter. My letter was supposed to be support for Dr. Karen Everett, who said this heretical thing that if we want to describe those 20 people, then standard deviation is what we should use to say whether or not those means really were meaningfully different or not. Not some metric based on a standard error. In other words, how does this compare to where it might be in the population at large, using the standard error, which would be the standard deviation, modified using the square root of n mathematically. Right. So anyway, Dr. Karen Everett and others make a clear statement that the variability of results should always be reported using the standard deviation. Great, okay, and not the standard error. Others have agreed and disagreed, blah, 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 background, paragraph. No problem. Okay. I would like to add to the debate by stating the following. First, the statistic reported in any given paper should be used to make the correct inference. In other words, I was against mandating speech, because they were talking about, well, these are the guidelines. You should use this metric. It should be the standard error. They were trying to say we should mandate this in publications. You don't have a choice as an author to use the metric you want to. We want to say, mandatory
right.
standard errors. So I'm kind of saying,
and to be clear, we're our example here. We are talking about the difference between 2 groups.
Yes. And
Yes,
they are still saying mandatory. You should use the accepted methodology. You should use the t-test with repeated measures if required, or, if you're talking about multiple groups, then we're talking about ANOVA tests, etc. But you should use those, and inherent in those is the standard error. You should not use the standard deviation to talk about the meaning of the difference in your particular population that you've looked at, your subpopulation, your sample, I should say.
And the two means that you actually observed.
That's right. Yes.
Okay.
So this is in the case. This is the context.
Sure I'm keeping up with this.
So the context is in the case where I might, as an author, want to describe the actual meaning of the difference I've actually observed, not what I might suspect I would find should I repeatedly, randomly repeat my study with a different set of 20 individuals selected at random and randomized to group A or group B at random as well.
Right.
Completely different metric.
Mm-hmm.
So I'm saying, look, instead of telling us how we should speak, you hateful fascists, how about insisting that publications that make inferences are based on the correct statistic for so doing? If I want to say, this is where the mean of my 20 people that I have sampled falls with respect to what I might expect from any number of repeated measures of 20 people taken at random, then I should use the t-test, the f-test, the one with the standard error inherent in the mathematics. That's what I should do. And I shouldn't say that something is not statistically significant because it falls within the noise band of the standard deviation of the observations I've made. That would be inappropriate. However, if I want to talk about the difference between my 20 people, 10 in this group and 10 in the other, then I should not use a standard error to suggest they were really meaningfully different, because they almost certainly were not, because of that noise of the standard deviation. So that's kind of the thrust of my letter. So that's what I'm kind of saying in paragraph 2 here. And by the way, if people want to read this letter, I will actually put the citation underneath this video so people can go and read it for themselves. And then we get to the third paragraph where I was asked to, tone that down a bit and clarify, what might be a suggestion of what I was talking about with you just now. In other words, if you want to talk about your group, use the standard deviation. If you want to talk about the population at large, use the standard error. Use the appropriate metric, no problem. Be disciplined. Don't make conclusions that are not supported by your empirical data set or by the statistics that you have used to make that inference.
Yeah.
And what was missing from that letter by the time it was published was the third paragraph that was between the ultimate and penultimate paragraphs in the letter as it's published. And that's the paragraph where I talk about saying the reason for this being a problem in science is because often people without training in statistics are left to believe, by the way that these papers are worded and stuff, that on the basis of a small scale study of, say, 20 people, 10 in one group and 10 in another, that they found this really huge, meaningful, large difference between those groups with respect to outcome variable Y, whatever that is. And as such, the intervention is now absolutely indicated, and we should all go forth and do that. Be that take this thing or don't eat that thing, for example.
Right.
And that's where my problem is. And this journal made me take that paragraph out to get it published, because it's standard practice to use the standard error to make such a conclusion, and by tacit failure to state or delimit your conclusion to that's what might happen to the actual value of the mean in any given repeated conduct of the study, again by somebody else, it speaks in no way whatsoever about the meaning of that actual difference as that relates to the noise around that, i.e. the standard deviation of any given set of 10 people.
Right.
So.
Right. And in these situations, we're looking at something we did differently between these two groups. We're looking at some variable we manipulated that we cared about and what that did to that Y, to that outcome variable. And it's that we treated one group this way and we didn't treat the other group that way. And that's what we're interested in. And the noise that's going to be problematic there that we want to pay attention to is how those means are distributed in these groups so that we can tell, is one group truly different than the other outside of the random noise we would expect to see, given these two different circumstances? And that's completely separate from our population. We do still want to describe these groups and own up to who this can potentially be generalized to in our population. But we're not trying to make a comparison about these groups and their means to a population mean. We are looking at differences between the means of these two groups. So what I'm taking away from your story, Bart, is that you went with this thing foolishly called common sense, which is not so common, and thought about doing things in a context-dependent way, depending on what the question you are asking is, what the research question you are trying to answer is, and how you actually have to assess that meaningfully. And Big Brother Journal didn't like that very much.
They liked it not one bit. In fact, they did not like my green eggs or ham. They did not like it, Sam. I am.
It sounds like they are depriving me of the best paragraph of your writing here.
Yeah, they are. They did.
Oh my gosh.
Yep. So that kind of speaks a bit to the political consensus-based foibles of the conduct of peer review science, where my role and my understanding as a scientist is to challenge everything, to accept nothing at face value, to falsify my own hypotheses and those of others, because a hypothesis stands as long as it stands refutation. And if someone can reasonably refute a hypothesis, sorry, this is a dichotomous positivist approach, which means something is true until it's proven not true. And as soon as it's proven not true, just once, we're done. Finished with that. Now we can dismiss H1 and we can accept H0 by way of the hypotheses. The null hypothesis, false. So when someone takes two studies, or takes two populations, I should say, subpopulations, samples of 10 people, and let's say in the experimental situation on the post-test, there was a difference in the mean between the pre-test and post-test measures on that group, and in the other group they were the same, to show, yes, a true control group that didn't change, because we didn't change anything, you changed nothing, nothing changes, great. In the group that we interfered, our experimental group, the value of y went up. The value of the mean of y went up, and it went up statistically significantly. That's what I'm reporting. Here is the standard error that I found. It was 0.01, just for fun, for a number. Ergo, doing that thing, assuming that the value of y going up is a good thing, is indicated, and you can all rest assured that that's what you should now do for the rest of your lives, based on my short-term 2-week study or whatever. However, is it conceivable, Jacquelyn, is it possible, in the situation I've just outlined to you, that of those 10 people in my experimental group, 9 of the individual people in that group experienced a 10% loss against the value of the mean, which actually went up. And that mean went up because the 10th person was an outlier and their value went up by 120% or something like that relative to its baseline. Value went up. as I've described. The standard error is exactly as I've just described because of my sample size, etc. How does the reader of that paper know that that's what actually happened in my study?
Without further information, they do not. Without showing your standard deviation, they do not. Without you being a responsible researcher and first doing an exploratory data analysis and pulling that outlier out of that group, they do not know. And unfortunately, those things don't happen often. And the powers that be are keeping things in place where it's okay.
Well, I mean, the powers that be, the journals, the peer reviewers are more concerned that I don't say something heretical against the way we do things and less concerned about, can I have a look at your raw data, please? Because no one has ever asked me for my raw data when I've submitted a paper for publication ever. Me
either. I've actually offered it and...
No, God, we don't want to see that.
Ooh, that's messy.
Because then I'd have to actually look at it as a peer reviewer. We don't
actually know what to do with that.
Yeah. Here's something people don't realize often as well. In academia, I was asked to peer review a paper probably 10, 15 times a week, every single week that I was involved in academia as an academic, which is a workload. It's a job if I'm going to do it properly.
It is.
I don't have to accept it. Of course, I can refuse, but it's a peer esteem factor for your record, for your CV, for your, this is what I'm doing to justify my chair sort of thing. And it behooves you to accept a certain number of requests to peer review papers. What do you get paid to peer review somebody else's paper? Nothing. So here's what often happens. The peer reviewer gets a reminder from the journal a working week out from the deadline to have the paper back. Have you reviewed that paper you agreed to yet? Oh, shit. Okay. Fine.
Yeah.
That's peer review. I'm sorry. If that bursts your bubble, boys and girls, when you read a peer review paper and think, it's peer reviewed. Oh, sorry. That's what happens. Am I wrong?
Yep. It was on someone else's desktop for some period of time. And they probably picked out a few things so that they could say, except with minor revision.
Yeah.
So, on page 16, you didn't, I'd like to see some 95% confidence interval. Okay.
Yeah.
Carry on.
Good work. Love your work. Carry on.
Right.
Also.
And if you do, I don't know if you've experienced this, Bart. I bet you have, because at first, when you do these reviews, I would go all in.
Yeah.
And I'd be like, feedback, questions.
Yeah.
And they'd be like, well, the other two reviewers said it was fine. So
yeah, exactly.
It's going in. Yeah. So if you do actually point out flaws and things like that, they kind of stop asking you to do reviews.
Exactly.
Which is problematic.
So in actually doing the job comprehensively, responsibly, properly, really looking at the paper, which, as a bright, young, shiny new academic, let's do this. Let's really show that we're doing this properly. Number one, nobody cares because there's usually 3 reviewers and the other 2 said it was fine. So they toss your review out and accept the paper, despite the fact that you wrote quite clearly there were several quite major flaws with it. And secondly, the journal will stop writing to you and asking you to do this because they're like, oh, shit, really? We have to read through another one of his reviews. The thing's a thesis.
Great thing. How many times have you submitted an article and the journal asks you, who do you want to review this?
Yes.
So who would you suggest to review this?
Yes.
So you get to pick your
blinded reviewer.
Objective reviewer.
Yeah. And then you email your blinded reviewer and say, hey, I've just nominated you. I've just tapped you to review my paper. Yeah. If you want to discuss the paper, I'll come down the hallway if you like, and I'll sit in your office for half an hour and we can talk about it. You probably, you've probably got other things to do, but yes, blind reviewers. Absolutely. And then the journal dutifully removes your name and affiliate from the
right. Don't put your name on the title page.
No.
The person you suggested won't know it's your work.
Yes, yeah. Whereas, I could tell you that when I had classes of sometimes up to 50 students, and I'd set an assignment, when the due date rolled around and the assignments rolled in, they were blinded so that I could grade them as such. But I knew exactly which student had written exactly which piece of work. It was obvious to me because I speak to my students. I know who they are. I spent time with them. I got to know them. I got to understand the way they speak. And as a student, before you learn how to write dispassionately without the flowery stuff that gives you away, students write in such a way so that you know who's written that paper. And the same is true in academia. When somebody gets on a high horse about something, I had several bugbears throughout my career, several things that I was out of step with, the accepted narrative on. And every time I submitted a paper and said, here's the reviewers that I think it should be, because I knew they were sympathetic, it completely erodes the truth of what happens in science for people. They think that this is genuinely an above board, blinded procedure which is undertaken seriously and solemnly by peer reviewers, and it's, that if something is peer reviewed, that its veracity is good. So I guess we're here to kind of disabuse you of that notion.
Yeah.
So today's discussion has been around standard deviation and standard error and how those are misused in science to make an inference that is not supported. So let's have, from Dr. Jacquelyn, let's have a summary of what is the standard deviation, what is the standard error, how should they be used, to close us off as a kind of repeat thing. So get your notebooks out, boys and girls. Take some notes here. It's very important. There'll be a test on this afterwards.
Absolutely. Jacquelyn. Yes. So let's summarize what we said. Our standard deviation is going to be the noise that we talk about within our sample or within our samples. That is going to be a measure of dispersion. That is going to be the average distance from the observed mean in our sample. And when we care about mean differences between samples or time points, we care very deeply about that standard deviation, because without knowing that, we can't know the meaning of any mean differences. We can't interpret what a difference of +2 means if we don't know what the random noise around that mean was to begin with. Because +2 could just fall within that random noise, and then all that is random. It's not meaningful. We'd expect to see a number fall within that range. So it even if somehow we are calling it a significant difference, it is not. It is not a practically significant difference. So standard deviation, measure of dispersion, average distance from our observed mean within a sample. When we're comparing a sample to the larger population, we want to have an idea of noise in the sense of how far is the mean I observed likely to be from the actual true mean in the population. And in that case, we're talking about taking samples over and over again from our larger population and looking at how the distribution of the samples of the mean. So every mean that we collect, where does it fall and how is it distributed? What is the standard deviation of any of those sample means within that spread? And so in that case, we really want to be thinking about our mean compared to the full population from which that sample comes, and how close is our observed mean in our sample to what is really true, the really true mean of the population we care about. And as I think Bart demonstrated really well, and we talked about a lot, that measure, that standard error, is highly, I'm going to say contaminated often with sample size. It's a statistical inference that has a very outlandish assumption that you could ever get to the full population size. And so it's really inflated when it comes to large samples. It makes the sample size, the N is the denominator in that equation. So a large sample makes for a very small standard error. And so people are using standard error in studies where we are concerned with mean differences between samples. And that is going to inflate and increase the number of times that we're calling something a significant difference where really it's a product of noise. And so I think that's a decent recap without spewing and babbling more.
Very, very good. All right. So, excellent. The standard deviation is the measurement of dispersion of individual data points around the mean value for that set, that sample of data points collected. It's the thing that tells us whether one group is truly different from another group. And that's what most studies actually are of. And so that's probably, in my mind at least, the statistic that should be used in order to determine the difference between those two groups, rather than the standard error, which is a measurement of really a standard deviation around the mean values that they would be should we repeat our study again and again and again and again and again. And the standard error, while it informs us by convention on statistical inference, P equals less than 0.05, good boy, get that published. It tells us absolutely nothing whatever about the noise around that signal. Unless you are smart when you read such papers, and what you do is you take the error bar presented, the standard error bar they present, and you multiply it by the square root of n in the case of that study, and that converts it back to a standard deviation, and you look as to whether the tails on those standard deviations cross by more than about 5%. If they do, noise to signal ratio, invalid study. Off you pop. Do it again. But most people don't know to do that. There you go. That's what you should do. Take your standard error, multiply it by the square root of n, and that'll tell you what the real noise around the signal was in the two groups that we looked at.
Yep, and you'll be more advanced in your statistical prowess even than most journal editors. So take it and run with it.
There you go.
Boys, girls, and attack helicopters.
That's it. Exactly. You're welcome. It was our pleasure. If you want to see more of Dr. Jacquelyn, and I'm sure you do, don't forget to comment underneath, because Al Gore really does love his rhythms. Yes, he does. And
I do have an idea. I was thinking as we were going through this part that it would be really cool to think about something we want to study and talk through all the decisions that have to be made along the way to design that study, how to actually do it. Because I think, like we talked about in our first video, people who haven't had to carry out independent research don't recognize how many times a scientist can basically be like making arbitrary choices about things within a study. Could be interesting. I don't know. People in the comments could say, no, that would be terribly boring. Don't do that. But
that, of course, would be hate speech. So there we go.
And I'd have to report them to HR. So
indeed. All right. Jacquelyn, thank you very much for your valuable time. Appreciate it very much. We do hope that you will come back and see us again.
Anytime.
Yeah. And once again, just to help you with pronunciation. Yeah, not bad for a girl.
Yeah.
You see what I did there?
Recovery.
Yeah. All right. Cool. Thanks for that. Appreciate it very much. I hope you enjoyed it, boys and girls, staff, attack helicopters, Lego trains, pine trees, whatever you are. I don't care. Generally. Yeah. See you next time, where probably someone will be wrong on the interwebs, because it doesn't look like slowing down anytime soon. There's still a lot of that going on. In fact, this week we've dealt with lame Norton once again, who, get this, Jacquelyn, actually said, and I thought, okay, there's a limit to stupidity, and vegans are generally tend to be, close to the hard end of stupid. And
Peak stupidity.
That's it. And we've heard them say this thing that I'm about to say that Lame Norton actually said as an argument. I never thought I'd hear it coming from Lame Norton. I know he's lame. I know he's a bit limited, the boy, but I really thought, okay, he's above this. Anyway, apparently he's not. He really said, get this, we can't possibly be carnivores, we humans, because we don't have sharp, pointy teeth and claws. Oh, really? God. Anyway.
He never got that spike in like brain development when we started using tools and getting certain nutrients and communicating with one another. So it's probably beyond his realm of understanding.
Yes. Anyway, that will probably be airing on the channel before this video. So you've probably already seen that. So that was probably no shock to most people. But anyway, that's how it is. Join me next time. And hopefully we'll be able to dive with ever more fervor into this murky pond of peer-reviewed science and why it's basically all a load of shit. All right. See you then. Ciao for now.